So we have completed prokaryotic transcription, and right now I'll be showing you animation of how that mechanism occurs. This animation is from the supplementary CD of Watson Molecular Biology of Gene, and we'll just have a summary of what we did, and then we'll move on to eukaryotic transcription initiation. So while talking about prokaryotic transcription, we had identified the main enzyme for prokaryotic transcription is RNA polymerase. And RNA polymerase is composed of several units that we also discussed. So this is how the RNA polymerase looks. It resembles a crab's claw. And this is how the RNA polymerase looks. We had discussed this. And you can see I had talked about an exit channel. And you can see the active site is found at this region. It's found at the base of the two pincers. So this is the active center cleft. And if you remember the mechanism by which DNTPs were added, it's very similar. The same mechanism by which RNTPs are being added. You can see this is something we had already done while studying DNA polymerization. So it's the same mechanism of RNA polymerization. Now, this is the transcription cycle we had identified the other day. So the polymerase, as you saw, got itself bonded at the promoter, and then it moves along the coding region of the DNA, and then after coding the terminator, it releases. So we have a phase of initiation, and the moment the polymerase escapes the promoter, we have a phase of elongation. Then we have a phase of termination, followed by the release of the polymerase and the nascent RNA chain. So this is also something we had studied. So in the first step, the RNA polymerase binds to a region called a promoter. And if you remember, we had discussed the bacterial promoters. And I had said that this particular factor called sigma factor helps in recognizing the various sequences within the promoter. In case of prokaryotes, we have two defined sequences in the promoter element, a sequence in the -10 region and a sequence in the -35 region. Another point to be noted here that the RNA polymerase with the sigma factor, yellow color is the sigma factor, binds the promoter in a defined orientation. So remember that the same DNA strand is transcribed always. So it is always the same DNA strand that is transcribed from a given promoter. And further remember, this complex, the polymerase has identified the promoter with the help of the sigma factor and is sitting on the promoter, and yet the double helix has not unwinded. So we are seeing that the double helix has not opened up. So this is the closed promoter complex, as I had discussed yesterday. In the second step, what happens? As you saw in the second step, the closed promoter complex undergoes to a transition to the open promoter complex. So while discussing the prokaryotic transcription initiation, initially what we saw, the polymerase bonded itself loosely with the -35 region, and the moment it attaches itself more closely with the -10 region, there is an opening of the DNA double helix for around 17 to 18 base pairs. That is what I had said. So you saw initially it is in a closed complex form. As the front of the enzyme further associates with the -10 sequences, you can see the change in position of the sigma and the DNA strand separates, forming of a bubble of single-stranded DNA. So this is known as the open promoter complex. Now, in contrast to RNA polymerase, if we look at the, in comparison, if we think of DNA polymerase, or rather put it the other way around, in contrast of DNA polymerase, if we look at RNA polymerase is able to synthesize a new nucleotide without the help of a primer. And this is what I had talked about, the promoter escape mechanism. So what you saw? You saw that RNA polymerase recognized the promoter, and there was a transition from closed complex to open complex. And then what you saw? The RNA polymerase is positioning itself in the promoter, and unless and until RNA chain longer than 10 nucleotides have been synthesized, the elongation phase cannot begin. So I'll go over this once more. See, the RNA exit channel is not open. Only when sigma changes its position, the exit channel opens, does the RNA polymerase escapes the promoter region. And the moment the RNA polymerase escapes the promoter region, what we see is that the sigma opens up, promoter escape has occurred, and the polymerase has entered the elongation phase. So once more, we'll go over this. You see the structure of sigma. The moment promoter escapes, sigma is opening and the elongation phase starts. So now the polymerase has entered the elongation phase. During elongation, as you can see, the polymerase unwinds the DNA in front of the enzyme, synthesizes RNA, proofreads the RNA, dissociates RNA from the DNA, and reanneals the DNA. You can see there is a comparison that is shown behind the enzyme. So you can see for replication, we had this host of enzymes. But when we think of transcription, is the single enzyme that is doing all the function. So it is unwinding the DNA, synthesizing the RNA, proofreading the RNA, dissociation of the RNA, reannealing of the DNA behind the enzyme. Whereas when we talk about replication, we saw that there is a helicase to unwind the DNA, there is a SSB to hold the DNA in proper position. There was a topoisomerase to unwind the DNA in front of the replication fork. So polymerase is able to perform all these functions without the assistance of other proteins. I'll go over this once more, see it by yourself. So the steps of elongation are going on. So polymerase is synthesizing the RNA, it's proofreading the RNA, dissociation of the RNA, reannealing of the DNA. Now you see the replication inset comparison. Now this portion is shown in the animation. So this is a simple mechanism of proofreading. We'll not get into the details of this. So if any by chance any incorrect nucleotide is added, that is corrected. And I had said you that DNA polymerase is more stringent, more stable in the sense that it makes a mistake of only 1 per 10 million nucleotides, while RNA polymerase allows 1 mistake per 10,000 nucleotides. This is because, as you realized, that DNA polymerase does not work alone. It has a host of partner enzymes, but RNA polymerase is the only enzyme doing all the job. There are 2 known proofreading mechanisms for RNA polymerase. One is the pyrophosphorylatic editing. Just the incorrect nucleotide is removed in a simple back reaction. So that RNA just slows down, and in a back reaction, the incorrect nucleotide is removed. So this is simple one. There's another proofreading mechanism which involves other proteins. So there's the hydrolytic editing. Another protein comes and removes the incorrect part. So the incorrect nucleotide is removed by additional protein. So the elongation phase continues. And ultimately, the polymerase reaches the terminator region. And as studied yesterday, we saw that there are two types of terminators: row-dependent terminators, row-independent terminators. We studied this yesterday. So first is row-independent terminators. So you can see the inverted repeats in the DNA, and corresponding to the DNA, there will be an mRNA synthesized. So polymerase is now transcribing the terminator region. The RNA has been synthesized, you can see, and the RNA forms a stem loop. You saw the formation of the stem loop. And yesterday I said, immediately the RNA forms a stem loop, and behind the stem loop was a string of U's, which helps the RNA to get detached from the DNA. This is case of row-dependent termination. So in this case, we do not have this two-fold symmetry. Rather, there is a Rath site. The rho factor binds, and the rho factor facilitates in an ATP-mediated manner the dissociation of the polymerase from the DNA, as well as the being a helicase in nature, it unwinds the RNA from the DNA. So this is in a nutshell that is being done in case of transcription. So you remember I said yesterday, the same segment of DNA can be transcribed multiple times. So in a given very small amount of time, we get a huge amount of RNA for a given DNA segment if that particular gene expression needs to be heightened. So multiple RNA polymerase, if you remember, the polymerase, the moment it enters escapes the promoter, the sigma factor opens up. So the sigma factor then binds with the fresh polymerase, sits on the promoter, and again starts transcribing the coding region. So a cell can synthesize, because of this factor, a cell can synthesize a large number of RNA transcripts in a very short time. So with this, we come to the end of our summary class. And so now we start with eukaryotic transcription. In case of prokaryotes, there is only one polymerase, while in case of eukaryotes have three different polymerases, and each category transcribing a different class of RNA. So that is the first difference between the enzyme that transcribes the RNA. Now, the second portion is that we have three categories of eukaryotic polymerases. Pol1, located in the nucleus, and transcribes the three major ribosomal RNAs, 28S, 18S, and 5.8S. RNA polymerase 2, located in the nucleoplasm. It transcribes the mRNA and some snRNA. So our enzyme, the enzyme on which we will be concentrating, where did my cursor go? The enzyme on which we will be concentrating is, just a second, this got hanged. So the enzyme on which we will be concentrating is RNA polymerase 2, because this is the enzyme that actually transcribes the mRNA. And RNA polymerase 3, located in the nucleoplasm, transcribes the tRNA, the 5S RNA, rRNA, and the remaining snRNA. Remember that Pol1 is located in the nucleus, Pol2 and 3 is located in the nucleoplasm. And RNA Pol2 of yeast has been shown to have 12 subunits. So this is about the polymerase. Now coming to the promoter regions. We have already studied water promoter elements. And we saw that in case of bacteria, there are two upstream element promoter elements, -35 and -10. When we talk about eukaryotic promoters are more complex in nature. There are two basic categories of promoters. The promoter analysis revealed two types of elements. One is the core promoter and the other is the proximal promoter. So if we imagine a eukaryotic promoter, from the start point where the DNA is being coding, if we move upstream, the upstream 200 base pairs adjacent to the initiator sequence is considered to be a eukaryotic promoter. The core promoter lies within the first 50 base pairs adjacent to the start site, and the proximal promoters lie from the remaining 50 to 200 base pairs from the start site. And let me show this by another picture, that picture. So if we have a start site, so this is in a transcription initiation, as you can see. So if we move upstream, so we have around 50 base pairs. We have around 50 base pairs upstream from the transcription initiation site. In this particular picture, they have included the transcription site and the downstream promoter element as core promoter. But if this is basically a picture of pairs, so this information varies a bit, differs a bit from the Russell. So since I have given you a study material related to Russell genetics, let's concentrate on the information of Russell. So then let me explain it with this picture. So if we have an initiator +1, so this is the transcription start point. So this is our transcription start point, and the first 50 base pairs. So this is our core promoter, and the next 50 to 200 base pair. So there is, as I said, around 200 base pair of sequences that forms the eukaryotic promoters. The first 50 base adjacent to the transcription start point are the core promoters. The core promoters also includes this +1. So there are two basic sequences, the +1 sequence, which is known as the initiator, which spans the transcription start site, and the -30. So we have a region here around -30, which codes for a sequence that is 5' TATAAAA. You see the sequence written here. And this is the famous eukaryotic data box sequence, or otherwise known as Goldberg-Hognes box sequence, after in the name of his discoverers. So the core promoter includes the +1 initiator transcription site and sequence upstream around -30 region, which is the TATAAA data consensus sequence, commonly or popularly known as the Tata box or the Goldberg-Hognes box. So just keep this in mind. These are the two main core promoter elements. And when we look into the region of the proximal promoters, we have another consensus sequence here. This is around -75 region, known as the CAT box. And we have another consensus sequence around here, which is the GC box. So promoter elements, when we discussed the promoter elements of prokaryotes, they were simple. There was a -10 region and a -35 region. When we compared with eukaryotic promoters, from the +1 upstream to 200 base pairs, usually the promoter elements are spread out. We have one very vital promoter element at -30, the Tata box, TATAAA. We have another important, that is the core promoter part, the first 50 base pairs, and the next 50 to 150 base pairs are the proximal promoters. We have two important sequences there, -75, the CAT box, and -90, the GC box. So this is the information about the eukaryotic. So we studied the eukaryotic promoters, and we observed that there are the two core promoters, +1 and Tata box, and there are the proximal promoters, the CAT box and the GC box. These are the well-known promoter elements. Now, when we talk about transcription initiation, if you look into this particular structure, we are having the Tata box. So this is our core region. So this is that Tata box area, and this is the +1 area. This is the site of transcription. Now we have to keep in mind that in case of eukaryotes, the initiation of transcription involves the assembly of RNA polymerase II and a number of other proteins which are known as general transcription factors on the core promoter. These general transcription factors first bind to the core promoter region and then recruit the polymerase to form a complex. So you see they are initially binding. So we are seeing that the general transcription factors are initially binding to the core promoter region, and then they are recruiting the polymerase to that region. So this is one thing that is important about eukaryotic transcription. Now, the well-known general transcription factors that are found here are TF2D, as you can see, TF2D, TF2A, TF2B, TF2F, TF2E, TF2H. So these are the different transcription factors observed in case of eukaryotic transcription. The general transcription factors are numbered for the RNA polymerase for which they work. So since these transcription factors are working for polymerase II, hence you see it's written as transcription factor Roman II. So if it was working for RNA polymerase I, it should have been written as TF, transcription factor 1, signifying that these transcription factors are working for polymerase I. If this transcription factor was working for polymerase III, then they would have been written in this manner, TF3. Since we are concentrating on the transcription of mRNA, so we'll again restrict ourselves to polymerase II, which is the main enzyme that binds with the template and transcribes the DNA. So all those transcription factors which are operative for polymerase II are thus named as TF2. So so TF2. And now coming to that A, B, C, D part. So in this example, D is the fourth, if you count A, B, C, D. So D is the fourth transcription factor that was discovered. So they are named in the order of the discovery, A, B, C, D. So D was discovered fourth in row that works with polymerase II. Now, for protein coding genes, the transcription factors, the general transcription factors and the polymerase II binds to the promoter element to produce the complete transcription initiation complex. So this is the complete transcription initiation complex. Prior to that, this is the minimal transcription initiation complex. So initially, minimal transcription initiation complex is formed, and then a complete transcription initiation complex is formed. Now let us come to this Pol2 promoters, as I said, contains the Tata element. We have talked about the Tata box. So this is our Tata element. This is where the pre-initiation complex formation begins. The Tata element, so this particular sequence is recognized by the transcription factor called as TF2D. Now, TF2D is a multi-subunit complex. The component of TF2D, so it's a multi-subunit complex, it has a Tata binding protein part and a Tata binding associated factors. So there are two parts to it. So there's a Tata binding protein and a Tata binding protein associated factors. So what happens? The Tata binding protein component of TF2 binds with the TATA sequence, TATA sequence, or the Goldberg-Hognes sequence on the DNA. And it also has another part known as the TATA binding protein associated factors, which serves as a platform. On this platform, you can see two other transcription factors known as 2A and 2B are binding. So we have a multi-protein complex associated now with the TATA element. Once the multi-protein binding complement has been situated on the TATA element, what happens, the moment the TATA binding protein binds with the TATA sequence, there is a distortion of the TATA sequence. And as I said, the TATA binding protein DNA complex provides a platform to recruit the other transcription factors. So TF2 and 2B comes. And now there is another transcription factor, which is the TF2F. TF2F then binds and TF2F, and what is TF2B? So we have TF2 and TF2B joined here. TF2F now brings the RNA polymerase and binds with this multi-protein complex already present at this TATA sequence to form the pre-initiation complex or the minimal transcription complex. Note here that in this minimal transcription complex, the promoter region has not yet opened up. So the promoter region is closed. Now, the conversion of pre-initiation to initiation complex is brought about by addition of two another transcription factors, TF2E and TF2H. Once TF2E and TF2H binds with the polymerase, you can see the opening of the DNA has occurred. In this case, TF2H has helicase-like activity, which facilitates or which stimulates the unwinding of the promoter DNA. So with the help of TF2H, the unwinding of the promoter DNA is achieved, and there now follows a short stretch of a period of abortive initiation, and finally the promoter polymerase escapes the promoter and enters the elongation phase. Once the polymerase has entered the elongation phase, the transition of polymerase 2 is achieved by its shading off most of the initiation factors that are attached to it. In its place, some new elongation factors bind to it, such as TF2S, and thus the elongation of the polymerase starts. And if we remember, we are studying the eukaryotic mRNA synthesis. So in case of eukaryotic mRNA synthesis, once the elongation phase starts and once RNA has transcribed some of the amount of RNA polymerase has transcribed quite amount of RNA, there is strikingly one thing that happens is the first modification of the mRNA, that is capping of the 5' end of the mRNA. So there is an overlap of the processing of the mRNA with the elongation of the RNA. So this is in a nutshell what happens in the initiation of eukaryotic transcription followed by elongation. We will not discuss termination in this class because, as I said, that eukaryotic mRNA is there are three categories of processing done in case of eukaryotic mRNA. There's a cap added at the 5' end. There is a poly tail added at the 3' end, and there is a splicing. Splicing is a different machinery, but the cap and the poly tail are important, and this processing occurs where the processing of this 5' capping starts immediately when the elongation is going on. So when we talk about the transcriptional processing, post-transcriptional processing occurring in mRNA, that is in your different topic. We will start with the 5' capping, And then termination is very much associated with the addition of the polyadenine tail. So when we'll be discussing the addition of poly A tail to the nascent mRNA, there only we will be discussing the termination of transcription in case of eukaryotes. So to summarize what we did today in today's class, what did we see? We saw that there are two categories of promoters, the core promoters and the proximal promoters. The core promoter has two important sequences. One is the TATA sequence and the other one was the +1 sequence. So these were the two important sequences. The TATA, the Tata box is situated at -30 region. In case of eukaryotic transcription, the enzyme involved is Pol2 because we're concentrating on mRNA, and the proteins are known involved are the general transcription factors. The general transcription factors are named for the enzyme for which they work. So the Roman 2 symbolizes that it is working for polymerase 2. They are named in their order of discovery. So transcription factor 2D was discovered fourth. Now, transcription factor 2D has two parts, Tata binding protein and Tata associated factors. The transcription factor 2D, the Tata binding part of the transcription factor 2D recognizes the Tata elements, and on binding with the Tata elements locally distorts those sequences, helps in, and the other part, the Tata binding protein associated factors also gets bonded to it. It provides a platform. Next comes TF2A and TF2B. Next comes TF2F and TF2F, which holds the polymerase and assists in the sitting of the polymerase in this multi-protein complexes. In this condition, the DNA is still not opened up. So this is a closed promoter complex, and this is known as the pre-initiation complex or the minimal transcription initiation complex. Next, TF2E and 2H join to this complex. TF2H is an helicase. Upon joining of TF2H, the DNA is unwounded and we get this open promoter complex. Now the polymerase is ready for transcription and it starts transcribing after a short stretch of abortive initiation and finally escapes the promoter. Once it escapes the promoter, it shades most of its initiation factors and fresh elongation factors are joined to it, and thus it enters the phase of elongation. When it is passing through the phase of elongation, then there only itself the 5' methyl capping of the hanging mRNA is done. Termination is associated with the polyadenylation of the mRNA tail. So we end our class here today.